I'm Bill Andrews. I'll introduce myself a little more during the talk. But it's probably everything else here. I'm here to talk about anti-aging. But I want to start off with first acknowledging the fact that a lot has already been done to make us all feel younger, look younger, healthier stuff. It's these people here, they've clearly been exercising a lot, seeing the right anti-aging doctors, eating the right foods, taking the right supplements. 65 is truly the new 45. And this is a good example. But I want to point out that I'm a little bit different from everybody else. When I turned 65, I want to be this. And so, in case you were wondering what talk you came into, this is what I'm talking about. I'm obsessed with finding some way to be like this. So, I want to talk about the fact, let's see if I can get this thing working. The question is, do we really have to age? And we all age, of course, but it turns out there are life forms on this planet that have now been identified that have no detectable aging process. That includes lobsters. People only started asking this question maybe about 100 years ago, how long do animals live? And they're finding that some of these animals that they've been watching are showing no signs of aging at all. And that includes lobsters, it includes tortoises, clams, some whales, some fish. Do you know that Charles Darwin's pet tortoise, Harriet, just recently died at 180 years old? And she's not the oldest tortoise on record. There are tortoises that are 200 years old that exist right now that people have documented. And there was a whale that was found that had a 130-year-old harpoon stuck in its side. So that whale had to be older than 130 years old. And then a clam has just been identified to be 507 years old. And unfortunately, when the scientists were actually measuring the age of this clam by counting its bands, they actually accidentally killed it. So this is just announced just 2 weeks ago. But they assured everybody that there are clams a lot older than that down in the ocean. Okay, so there is a cell type. What I'm going to be doing today is I'm going to explain why are these animals all different from the rest of the animals on the planet. Okay, what makes them different? But I want to point out that there is a cell type in us that doesn't show any signs of aging, and that's our reproductive cells. If our reproductive cells were to age, then our children would be born as old, if not older than we are. So something is going on in our reproductive cells that keep our reproductive cells from aging. We've been passing on these cells from one generation to one generation since the beginning of humankind, and they're not aging. So why is that? And that's one of the things that I got into this field 20 years ago, asking that question. So I'm Bill Andrews. I'm the president, founder, and CEO of a company called Sierra Sciences. Our motto is cure aging or die trying. And we're located in Reno, Nevada. I've been in biotech for 32 years, focusing mostly on cancer and heart disease and inflammation research, because I was always saying to myself, there's got to be some kind of clock that explains the aging process. The environment kind of stories never made sense to me. Why do people who live on the North Pole and the South Poles age the same way as people who live on the equator, if the environments are terribly different? And why do cats and dogs age at different rates than humans when they're in the same environment? So I decided there had to be more than just environmental impacts. There had to be some kind of clock that ticks inside of us. And it took me until about 20 years ago, until I was attending a conference, I heard somebody else talk about something that could be that clock. And I've been hooked on that ever since. I have 45 U.S. issued patents on the subject that I'm going to be talking about today. And for the discoveries that I made, I was awarded second place for United States Inventor of the Year back in 1997. And the subject that I'm talking about is just called short telomere disease. I used to call this aging, but as you'll see, it goes way beyond just aging. We all suffer from this. We're all the less fortunate. We all have short telomere disease, and what can we do about it? So to explain what a telomere is, now sometimes I find audiences, there are still people who don't know what telomeres are, so I'd like to show this, but sometimes I find out there's audiences that people think they know it and they still don't know. So I go through this kind of basic background on what a telomere is, at all my talks right now. But to understand what a telomere is, we need to zoom in on a human being. We find out that a human is made up of 100 trillion cells. And most of the theories on why we age say that we age because our cells age, and so we got to figure out a way to prevent those cells from aging. Let's zoom in even further. Remember, we're trying to find out what a telomere is here. We find that every cell contains a nucleus, and inside the nucleus are found the chromosomes. They're, of course, where our genes are that give us our hair color, our eye color, and a lot of other different things. And they're all organized on a DNA molecule that extends from one end all the way to the other. And DNA is like a long string of beads. The beads are called bases. And a typical DNA molecule in a chromosome is about 100 million bases in length. Okay, so let's focus on what's at the very tips of these chromosomes, because that's where the telomere is. Now, if we unravel, the DNA is all kind of folded up like a slinky inside this chromosome. And if we unravel the slinky at the telomere, we find that the telomere is about 15,000 bases in length. Remember, I said that the chromosome is actually about 100 million bases in length, so the telomere is pretty small. I want you to think of a shoelace. Okay, chromosomes like a shoelace, except wound up like a slinky, and the caps on your shoelaces are the same as the telomeres on your chromosomes. So I said that they were 15,000 bases at least when we're first conceived. But here is where the whole problem is. As soon as our cells start dividing, the telomeres get shorter. Every time a cell divides, a human cell divides, the telomeres get shorter by about 50 to 100 bases. And so as a result, from all the cell division that occurs from the time we're conceived to the time we become a newborn baby, our telomeres have shortened from 15,000 bases down to 10,000 bases. And that's bad news, because we don't really want telomeres to be shortening. Okay, so now we're going to have to grow up now. Okay, there's going to have to be more cell division, and as a result, our telomeres are going to get shorter and shorter still as the, I guess it didn't go for there. Yeah, as the cells divide, the telomeres get shorter and shorter and shorter. And we know from working in the lab and looking at people's skin, when telomeres get down to about 5,000 bases, they lose the ability to function, and that's when we die of old age. Really, really old people have telomeres that are about 5,000 bases in length. So let me go over that again. We're conceived at 15,000 bases, we're born at 10,000 bases, and we die of old age at 5,000 bases. And there's absolutely nothing we can do about that, at least yet. Okay? No matter how well we eat, no matter how much we exercise, no matter how much we do everything our doctors tell us to do, or we tell ourselves how to do, because there are a lot of doctors in the audience, we can't stop this shortening. Remember, I was saying I was looking for a clock, some type of clock that ticks inside of us that controls our aging process. This fits that clock perfectly. And so it's the only clock of aging, as far as I'm concerned, that's ever been discovered in humans, that when we find out if we manipulate it, actually affects aging in human cells and other types of organisms that are equally affected by telomere shortening. I can take blood from any of you, I can measure the length of your telomeres, I can tell you your biological age, and more importantly, I can tell you how long it'll be before you die of old age. Okay, and that's what's really key about what I'm talking about today. So I want to congratulate my colleagues that I worked with 20 years ago, Elizabeth Blackburn, Carol Greider, and Jack Sostak. They did the pioneering work. They won the Nobel Prize in Medicine in 2009 for doing the pioneering work that led up to everything I'm going to be talking about today. They showed the importance of telomere shortening. They showed how there has to be some way of lengthening telomeres, and that was really important. And they won the Nobel Prize for elucidating all that, because that led to all the research that me and other people did to really figure out what's going on here. Now, I was awarded second place for United States Inventor of the Year for figuring out a way to stop the shortening and actually re-lengthen the telomeres. And also, I mentioned before that our reproductive cells show no signs of aging. Well, it also showed here what was actually causing our reproductive cells not to age, and it was the lack of telomere shortening. Now, because of this, thousands and thousands of publications started coming out right away as soon as we figured this out. And now practically every disease you've ever heard of has been connected to the length of your telomeres. Telomere shortening is bad for you. I always say bad things happen when telomeres get short. Okay, there's nothing wrong, there's nothing good about a short telomere, but there's all kinds of things, there's all kinds of good things about long telomeres. Nothing wrong with long telomeres. I'm not going to go into these diseases today because of lack of time, but I can talk about any of these things if anybody's interested in hearing more about it. You can also read, Popular Science did an article about me and my research recently. You can read about that. I'm also coming up in an HBO documentary in first quarter of 2014. You can listen to that and learn more. I also have a book that's coming out. It's not available yet, but it'll be out soon, where you can learn a lot more about telomere biology. It's written mostly because there isn't any place else that really goes into telomere biology in great detail. And then lastly, I want to point out that since we're in Nevada, the governor of Nevada recently declared September 2nd through 8th as Telomere Research Week. It's a proclamation that was actually signed by the governor because they are really excited about what we have to promise here. And, you know, we're probably going to put Nevada on the map because of the discoveries we're going to make here. Okay, so what I want to do is I want to talk, this is pretty much my introduction there so far. What I want to do is I want to talk about what can we do about telomere shorting. There's pretty much three things we can do, and usually I could talk hours on each of these things, so I'm not going to talk about all of them in great detail, but today I want to focus on know the length of your telomeres. This is a new technology. People are getting, there's places out there right now where you can have your patients or your own telomere lengths measured and see what it's telling you about your overall health and your lifespan, etc. Okay, so what I want to do is I want to show that first, if you take human cells and grow them in a petri dish, and you measure their telomere lengths all through their lifespan, you can find that you get a pretty accurate straight line. Okay, there's telomere shortening, telomere length is very correlated to the number of times a cell divides. So the more cell divisions, the shorter it gets. You get a pretty nice straight line. Now sometimes when you take human blood and try to measure the telomere lengths there, you get a fairly decent line too. But more often than not, you get something that looks like this. Okay, so telomere length measurement has its flaws right now, and we're not exactly certain 100% what's going on here. But one of the things that I recommend, I think, is really think about what it is that you really want to measure. Okay, do you want to measure the average telomere length, or do you want to measure the percent of telomeres that are short? I want you to think of that shoelace. Actually, first let me say that there is a paper that actually came out recently saying that the shortest telomere, not the average telomere length, is critical for cell viability and chromosome stability, saying that the health of a cell is more determined by the number of short telomeres you have than it is your average telomere length. So think of that shoelace again. If a shoelace, when the caps all fall apart, your shoelaces fall apart. But if your shoelace gets half the length, it's just as good as it was when it was full length. Okay, the problems all start when your shoelaces start getting so short that the caps fall off. Okay, and that's what's happening in our telomeres. When our telomeres get really, really short, our chromosomes start to fall apart, and we have chromosome rearrangements and mutations and stuff that cause all the diseases that we suffer from. So again, more research is done right now, but right now everything is pointing, as far as I'm concerned, to the idea that we want to be looking at the percent of short telomeres. So if you have a patient and you want to send them out to get their telomere lengths measured, I recommend, I'm going to say more on this, you recommend that you look at the percent of their telomeres that are short. That's much more important than looking at the average telomere length. Okay, there's something else here about telomeres. Okay, nobody still knows why the length of a telomere has anything to do with aging. It's just a correlation. We've found that we lengthen them, cells, petri dish, and I'll show more data later saying that cells actually get younger, okay, and healthier. But here's one model to kind of address why that is, okay, and it will also explain why, again, it's more important to be looking at the abundance of short telomeres. This is a chromosome. The green, the red, and the red, think of those as genes. Green means a gene that's being expressed. Red is genes that are repressed. Okay, the telomere is off at the far right, according to you. I think of the telomere as something that actually folds back onto the chromosome and turns one of those red turned off genes into a turned on gene, but just by touching it. There actually is a precedence for this, for those of you that know about gene expression and know about enhancer sequences that can be like 10,000, 100,000 bases away from a chromosome. DNA actually folds over and does play a role in turning genes on and off in the rest of our chromosomes. So why not in the telomere too? So it could be turning this gene on, it could be turning that gene off, it could be turning that gene on. But when telomeres get short, they can't fold over anymore. So the telomere can't reach these genes anymore. And so this might be a clue as to how telomeres are affecting our cells, what's going on inside of our cells. There are several different protocols that now exist for measuring telomere lengths. We do almost all of these in my labs. What's important is that the first four here, they look at average telomere length, and the bottom two are the only two that actually can look at the percent of telomeres that are short. And what I want to say is that, because if you're sending your patients to get their telomere lengths, there are several places that commercially offer this, but only one of these is actually able to provide you with percent of telomere shortening, percent of short telomeres that's a commercially available thing. So I just want to let you know that such a service is available. Okay, so next I want to talk about, not as much time as I'd like to talk about if you have a patient and they have really short telomeres, what do you do about it? Okay, because a lot of people say there's nothing you can do about it, but I believe there's a lot you can do about it. And I just want to go through a bunch of things. Exercise. Intense endurance exercise has been shown to be a good way to reduce the rate of shortening of your telomeres. People who are sedentary, people who run 10Ks, people who run marathons, people who run ultramarathons, have all been looked at, and there's a very great straight line that shows the more intense your endurance, the longer your telomeres. Not because your telomeres got lengthened, it's just they decrease the rate of their shortening. Since I'm an ultramarathon runner, I'm really glad to hear that, because I think I've been doing the right thing all my life. And I'm actually 87 years old, and people say I look no older than 60. All right. Antioxidants, omega-3s, and vitamin D, those are also things that you can do to decrease the rate of telomere shortening. I highly recommend those. Smoking generates free radicals and causes inflammation. Both those things cause what I call accelerated telomere shortening. This is why people that have been smoking all their lives look older than their friends that didn't smoke. Obesity, same thing. Obesity causes accelerated aging, generation of free radicals, inflammation. Stress. Now there's a few things here that have been surprising. The Nobel Prize winners have published a lot on this recently. There's been 2 studies that looked at caregivers of Alzheimer's patients, not the Alzheimer's patients themselves, the caregivers, and found out that they had shorter telomeres than their friends their same age because of the stress this caused from taking care of somebody. So if you are in a position like that, you got to get out and take care of yourself, okay, because you're accelerating your aging and you're accelerating your decline of health. Depression. Depression's also been shown to cause accelerated telomere shortening. A little harder to control, but there's ways to controlling that. I want to mention on stress, back up here for a second. There's also been a study showing that adults that were abused as children have shorter telomeres than adults that weren't. So there's a lot of connection right now showing that stress and depression cause accelerated telomere shortening. Pessimism, surprisingly, there's been 2 studies now showing that if you're in a room and they ask everybody, do you think you'll live to be 100? And then they measure all your telomere length. It's been repeated twice now that people who think they won't live to be 100 are right. They have shorter telomeres than people that don't. But one thing that doctors now that are practicing telomere biology in their offices, they're telling all their patients, the key thing is be happy. This is a really good thing that correlates with telomere shortening. So just enjoy life, reduce stress, reduce depression, all this kind of stuff. And the good news is these are all things that you can do now. So when you get your telomere lengths measured, or you get your patient's telomere lengths measured, if they have a short telomere, then give them all these things to do. They've got to start practicing all these. Okay, next I want to talk about what we can do to actually increase the length of our telomeres. And this is a brand new science. It's only come in the last 20 years. And it comes from the fact that we know that telomeres don't shorten in our reproductive cells. I said that's how I really got into this, trying to ask that question, why is that? And I led the research that discovered this enzyme called telomerase. This green little squiggly thing is the DNA molecule shown as a double helix. and telomerase is bound at the end, and it's lengthening the telomere. But it's only found in our reproductive cells, at least in normal human cells. It's found only in our reproductive cells, and they lengthen the telomeres. So it's like, think of it a clock. Every time the clock ticks once, that's every time the cell divides, the telomeres get shorter. It's like ticking a clock once. Telomerase actually pushes back that tick. Ticks again, it pushes back that tick. So the telomeres actually don't get shorter in our reproductive cells. Okay, and as I said, it's only found in our reproductive cells. But what would happen if we caused telomerase to be produced in all the cells of our body? Well, that's exactly what happens in lobsters. Okay? People knew that they were seeing no signs of aging. It was published that telomerase is produced in every cell of their body. Okay? Now, there's a lot of people back 10, 15, 20 years ago that used to say, boy, if you produce telomerase in all the cells of the body, that's going to give everybody cancer. We now know that's not true. Most studies are saying that actually telomerase lengthens the telomeres and decreases your risk of cancer and increases your body's ability to fight cancer. But these lobsters, they have practically no detectable cancer, even though they have telomerase turned on in all their cells of their body. The same is true for all those other animals I mentioned, the tortoises, the whales, the clams, the fish. They all have telomerase produced in all their cells of their body, and they get low incidences of cancer, and they have no detectable aging. So what my mission is to make us all like these animals here. We all want to be like them. My company is in Reno, Nevada. We've been working there since 1999. We're searching for telomerase inducers, things that will cause the induction of telomerase inside of our cells that stop or reverse telomere shortening in humans. That's the number one only goal of my company. This is just some pictures of my company with some of the scientists working there. And this is our strategy that we're working with. This is, the gray bar is the chromosome. And like all other genes, there's a gene that produces telomerase. Okay, now next to all genes, there's a regulatory element. And this is like a light switch. It turns the gene on and off. And so we all have, for those of us that have advanced molecular biology, I'm talking about promoters here and transcription factors and things like that. But for the sake of this audience, there's a regulatory element next to every gene, and it involves in the regulation, the turning the gene on or off. So in our reproductive cells, the telomerase gene is turned on. In all the other cells of our body, there's a repressor protein. It's called a repressor that binds to that regulatory element and shuts it off. So in all the other cells of our body, we have no production of telomerase, and therefore our telomeres shorten. So what my company is doing is we're looking at synthetic chemicals, pharmaceuticals, natural products, just about anything, looking for anything like this green thing that will bind to that repressor, dislodge it, and allow the gene to turn back on again. And we've been doing this for 15 years now. We've actually, and think of it like a dimmer switch, because some of the green things will turn it on a little, some will turn it on a lot. So it's not just a flip of a switch, it's like a turn on the knob to get more and more telomerase production. We've actually found quite a few. We've found like 900 different chemicals right now that turn on telomerase. We've been able to break them down into 39 different families. So be prepared for this in your practices. There's going to be some really good things coming along. Our highest so far, we call it 16%. That means it is produced. We have some chemicals that when we add to human cells in a petri dish will turn on the telomerase gene to about 16% of the level needed to actually make that cell immortal by scientific standards. So it makes it so those cells show no signs of aging. And we're at 16% now. We think we're going to be at 100% within 1 year. And then we think we'll be ready for human testing within 3 years. So we're really close on this stuff. And we think we're going to have skin creams, pet products, cancer diagnostics even sooner than that. So a lot of stuff is going to happen in practices right now. We are just a research company. I have no idea where you're going to be buying these products or anything from that. We're just research trying to figure out these things. But I want to say that there are natural products already on the market. And before I actually get into these, I want to talk about another study that came out showing that short telomeres are preferentially elongated by telomerase in human cells. This is important because the natural products are relatively weak, and so to be able to look to see if they're actually lengthening the telomeres, we have to actually look at the shortest telomeres. So again, we're getting back to it's important to measure the percent of telomeres that are short. It's more important than measuring the average. And I want you to think of a telomere in this sense to explain why this is. Why does a short telomere preferentially get elongated? Think of a telomere as actually wound up like a ball of yarn. This is the chromosome, and then the big ball there is the telomere. Well, the tip of the telomere is found inside that ball of yarn. No data is supporting what this is just a theory here. But the telomere is found inside of that ball of yarn, and as a result, telomerase can't get into it to actually bind to it and start lengthening it. But when telomeres get short, telomerase can gain easy access to the telomere and lengthen it. So when you produce low levels of telomerase inside of a cell, you're best to be able to look at the short telomeres because that's where you can detect to see if there's any lengthening. And if there's lengthening, you know there's telomerase there. So one of the natural products was published as a clinical study. I'm actually one of the authors on it. And what I want to show is that, again, the question that they ask in this paper, what's more important, measuring the average telomere lengths or the percent of short telomeres? And so here's data where they looked at the average telomere lengths of 12 different people. These are people that were taking the natural product telomerase inducer. The black bars are before they started taking the telomerase inducer. The gray bars are 1 year afterwards. And the y-axis here is the average length of their telomeres. And what you can see is that out of the 12 people, 6 of them saw a lengthening of their telomeres, which is good. But the problem is that 6 also showed a shortening of their telomeres, which is making this completely random. And what I'm trying to tell you is that measuring the average telomere length right now is not something that's very productive because it's so far removed from what you really should be looking at, and that's the percent of short telomeres. Okay, so they also looked at the percent of short telomeres here. And again, the same 12 people. And what you see in this case is that 10 out of 12 saw a lengthening of their shortest telomeres. And only 2 saw a shortening. And it turns out those 2 were the youngest people in the study that had so few short telomeres to begin with, it was very hard to measure. So it's a really good data here, very strong correlation showing that if you were looking at something, a weak telomerase inducer, if you're trying to have a patient, you want to help them by extending the length of their telomeres, it's best to be looking at the percent of short telomeres. So bottom message here is when measuring telomeres, measure the percent of short telomeres. Okay, so now what I want to do is I want to spend some time talking about the future because I think the future is very exciting in this field of telomere biology. There was one study that just came out of Harvard where they using telomere lengthening technology were able to lengthen telomeres in old mice. The title of the paper is not quite clear, but it's telomerase reactivation reverses tissue degeneration in aged telomerase deficient mice. This is actually very exciting. The press releases came out saying telomerase reverses the aging process. First time in the history of the planet that anybody has actually successfully turned old mice into young mice. And what they saw was a 33% increase in telomere length. So what they did here is they engineered these mice so that they could turn telomerase on and off at will. They allowed these mice to get really old. They lost the ability to reproduce. They could not remember how to go through a maze. A lot of their organs got smaller. Their hair was falling out. Their hair was turning gray. They couldn't run around. They were so old they were ready to drop dead. They then treated them with a chemical that will only work on these mice because these are engineered mice. They figured out a way to turn it on and off. But they turned the telomerase gene back on that lengthened the telomeres 33%. But they also saw these mice could reproduce again. They saw organs get bigger, their spleen size, their brain got bigger, their sense of smell came back, their brain size got bigger, but they were able to remember how to go through a maze again that they had forgotten how to go through. And I think that's exciting because it's a potential solution to the Alzheimer's problem. And they also saw a threefold increase in survival after 25 weeks. Now, So this is really exciting data. Is that hopefully this data is going to be reproducing. It's actually been reproduced in one lab in Spain, and where they didn't go to quite the same extremes, but they did see an extension of the lifespan. So what I want to just do is spend 3 minutes. I think I'm.
And now, eternal youth.
This is
it
a
in a
press release
cage around the corner? News tonight of a breakthrough for some pioneering mice. But we always wonder, what does a fountain of youth for rodents reveal for humans? Here's Sharon Alfonsi reporting.
I feel tremendous.
In the movie Cocoon, it's a swimming pool that turns back the clock for a group of senior citizens. But now researchers have found a way not just to stop, but reverse the aging process. The key is something called a telomere. We all have them. They're the tips or caps of your chromosome, seen here in yellow. This is what it looks like in a young adult. But as you grow older, the telomeres become damaged and frayed, and as they stop working, we start aging, experiencing things like hearing and memory loss. Scientists took mice who were prematurely aged, added an enzyme, and essentially turned their telomeres back on. You can see it before the enzyme, after.
Their brain function improved, their fertility was restored. It was a remarkable reversal of the aging process.
Look at this picture. The mouse on the right has bad skin, gray hair, and is balding. But the one on the left had its telomeres flipped back on.
And you could see that essentially you now have a dark coat color, that the hair is restored, that the coat has a nice healthy sheen to it.
Even more dramatic, the change in brain size. This is before. The mice had 75% of a normal brain, like a patient with severe Alzheimer's. But after the telomeres were reactivated, the brain returns to normal size. As for humans, while it is just one factor, scientists now say by looking at our blood cells and measuring those telomeres, you can get a better idea of how well you'll age. The longer the telomere, the better the chances for a more graceful aging. But as for tinkering with them and turning back our aging process, researchers say we still have a long way to go. Sharon Alfonsi, ABC News, New York.
Well, as I said before, we don't have a long way to go. I think it's going to be a lot sooner than everybody expects. And I think it's exciting. We've now figured out how to turn old mice into young mice, and humans are next. Thank you very much. And I was hoping to leave 15 minutes for questions, but there's still 10 minutes. So questions, yes?
Is that natural supplement available? Where do we get it?
I'm not allowed to talk about products here at this conference, but there's two booths. Actually, there's 2 products at least. There's 3 I know of. There's 2 here at the conference. So at the expo, yes?
You say that reproductive cells don't have the repression gene turned off.
Yes.
Well, from conception then, what time period in the human development do those other cells develop and differentiate that repressor cell?
It's about the 8-cell stage. So at about 8 cells, an 8-cell embryo, 7 of the cells suddenly shut telomerase off, and 1 of them goes on to be our germline. Okay. Other questions? Yes?
I have a question. One of them is there really any significance to the average telomere length? The measurements in front of people with the average length are really long, with the short telomeres which are appropriate for their age. I mean, is there any relevance to that?
I think the average telomere length is a little easier to measure. It's a lot cheaper to measure. It's not that meaningful, but I think it's just a weak biomarker for what you really want to measure, which is the percent of short telomeres. So when you increase the number of percent short telomeres, your average is going to go down just as a consequence. So it's not as specific, but there's a correlation. I just think I personally, in my research and stuff like that, am much more interested in, I see much better correlations between like the number of times the cell divide based on the percent of short telomeres. Yes, back there.
What happens to the telomeres in rapidly producing cancer cells?
Okay, so telomeres, when telomeres get really short, that causes a lot of chromosome rearrangements and mutations. And those chromosome rearrangements and mutations are what causes the cancer. So we don't want to let our telomeres get short. In fact, this is up to like 5 years ago, there wasn't really any data on this, but now there's hundreds and hundreds of peer-reviewed, scientifically peer-reviewed journal articles showing that people that have cancer, their cancer cells have really critically short telomeres. The telomeres got short first, and then they became cancer. And also their telomeres and their immune cells are weak. So one of the main causes of cancer is a weakened immune system. So we want to keep the telomeres long in our immune systems to fight the cancer. So personally, if I had cancer, I'd be doing as much as I could to lengthen my telomeres, to decrease the chances I get other cancers, and also to increase the chances that my immune cells can fight the cancer. And a study just came out recently that I thought was very interesting, is that it's now been shown that the cells in women's breasts, all women, not just the ones that have breast cancer, all women have, the cells in women's breasts have shorter telomeres. And this is probably why women have a higher incidence of breast cancer than men do. But, you know, it requires more cell divisions in a woman. But I think it's a lot of data still coming out. I think the same is going to be shown for prostate cancer pretty soon, but it's a brand new field. Yeah, so he's got one more. Okay.
My question was, do the telomeres get shorter as these cells are dividing?
In cancers?
In cancer?
No, they don't. They stay at a critically short level. Okay, you go ahead.
It's a big jump from 16% to 100% in 1 year. Do you have any heads up on why, what's happening there?
Okay, so we started doing random drug testing, or synthetic chemicals. We got up to 6%. Then we started doing what's called medicinal chemistry, and in 2 months we went from 6 to 16. We think we can go from 16 to 100 very easily. So we've hardly scratched the surface. So going from 16 to 100 should be pretty fast, and we think we'll get beyond 100 even before then. So yes.
Do you think there's any risk that telomeres shortening is a result rather than a cause of aging?
That's what used to be thought. Now everything points to the fact that telomere shortening is a cause of aging. There's, of course, declining health things cause free radical productions and inflammations that could also cause the telomere shortening. But it's just the simple fact that we can lengthen telomeres and make human cells in a petri dish, human cells grown into skin on the back of a mouse, all kinds of studies showing that the cells actually become younger by every method of measurement we know of. Yes?
Do cancer cells not produce telomerase?
They do produce telomerase. One of the mutations that occurs, as soon as you start having all these mutations, one of the mutations that occurs is to turn on the telomerase gene by a mutation.
But then it's too late.
But for the cancer cell, yeah, the cancer cell. So you want to do it before the telomeres get short. You want to be lengthening the telomeres before they get short. Now, in no cases, maybe 1% of the cases, maybe the telomeres might actually get a little bit longer in the cancer cells, but in most cases, it's low levels of telomerase that just barely keep the telomeres long enough. So this is why some of the therapies, in fact, I'm a named inventor on some of them, where we actually inhibit telomerase to cause those cancer cells to die of old age instantly. Yes?
Is there a known percentage of short telomeres at which it makes the most sense to introduce therapy to prevent further shortening, or would this be universal? We should start taking supplements at age 20. I
believe that unborn children should be getting their telomeres lengthened. I think telomeres. There's nothing wrong with long telomeres. It's just you want to keep them long, and just from the very day you're conceived, you should be doing something.
Well, if
yes.
It's a universal good, then why bother testing? Why not just treat everybody?
Okay. It's
You're okay with that?
Because, well, so far there isn't a technique for really lengthening them. Okay, only we're at a decrease in shortening. But the same thing could be said for every kind of cholesterol test or anything like that. But you're doing everything you can to keep your body healthy, but nobody does. Okay, so we all want to get measured, and then when you see that something's wrong, that's when you get yourself back into gear. Yes, back there.
We were talking about lack of telomerase activity on somatic cells. How about stem cells?
Well, the stem cells don't have telomerase. We can detect low levels of telomerase in stem cells, okay, but it's so low that it's actually a result of the rapid cell division causing leaky expression of telomerase, not actually a planned thing to have telomerase produced that lengthen telomeres. When you take stem cells out of a gut or a bone marrow and then culture them outside the body, the telomeres get shorter. When you inject those stem cells back into the patient, those cells are older than the patient, even though they came from that patient. And so that's why a lot of people who have done bone marrow transplants or skin transplants start finding that their tissues start dying of old age 15 years later, even though they're still young. Back there. Yes. I can't at this conference. It's against CME rules. Yes.
So I'm still a little confused on the reproductive cells that have telomerase, and some of these companies are saying there's products that turn on telomerase in your body. Is it only working on my reproductive cells then, or what's that?
I didn't understand. Okay, repeat the question.
So which cells have telomerase?
Only reproductive cells.
Like sperm and
Yes, the cells that make the sperm and the eggs.
So you get with telomerase activating supplements. Is it only activating it?
No, it's activating it throughout the body. Yes.
Turning it on.
Yeah. And actually, answering that question back there, I can't tell you the product, but look around for me. Okay, I'll be at their booth. Okay. Yes.
So the products you mentioned, is it just shorten it shortens the time period of the telomere shortening, or does it actually increase the length of the telomere?
Okay, so telomere shortening is like a tug of war. You've got shortening and lengthening going on at the same time. Okay, so so the first things I talked about, like the exercise, the smoking, the things like that, that actually causes more people to be on the tug of war side that's shortening the telomeres. Okay, so telomerase inducer, the supplements, are pulling on the side to lengthen them. But they're not strong enough yet to outdo the shorteners. So still the net result of the supplements that exist right now is that the average telomere length still gets shorter, even though the shortest ones can get longer. Yes?
I work with a doctor who does fetal stem cell therapy, and he has had great results in terms of increasing telomere length, which has resulted in reversing conditions like Alzheimer's and other kinds of conditions. Would you like to comment on that?
Well, some people are making that work, but are they human embryonic stem cells when you say fetal?
They're human fetal stem cells.
Okay, so I guess I'm not up on, I've published a lot on stem cells, but I'm not quite certain what you mean by human fetal stem cells.
I'd be glad to talk to you.
Okay, yeah, let's talk. But human embryonic stem cells, they have telomerase turned on. They're the only natural cell that has telomerase turned on. They're part of our reproductive cells. Yes, back there.
Any studies on consuming the active enzyme versus activating?
Yes, I mean, indirectly, there's so many, there's companies out there right now telling you that they have a cream or something like that with telomerase in it. But telomerase is too big to get inside the cells. And you have to actually produce the telomerase inside the cell in order for the telomerase to be there. Telomerase is it's a big protein with an actually RNA component involved too, and it's just way too big to be taken up by a cell.
So it's an absorption issue.
Yeah, it can't be absorbed by the cells. It's too big. Yes, Dave.
qPCR is a very common way of measuring. It's very common in all the studies. A lot of studies use that.
Average telomere length, there is some good things about them. I mean, it does work for large population studies. If you want to test 100,000 people, and one company just did that with Kaiser Permanente, patients at Kaiser Permanente, average telomere lengths will tell you if something is affecting telomere shortening. Because even though there's a lot of variation, the variation gets canceled out by the large number of samples. But I'm trying to talk about your patients, okay? All that variation isn't going to work for the individual. Each individual is going to be random. So a lot of the big studies going on can still be done with average telomere lengths, but even there, they're going to need a lot less, let's say, patients in their study if they look at the percent of short telomeres instead of average. Was there a question back there? Okay, you're just scratching your head. Yes?
From an evolutionary perspective, what is the advantage of having developed inhibitor of telomerase?
There's no evolutionary advantage to living longer than it takes to raise your young. After we've raised our young, we're just in the way. Okay, so it's important for the survival of the species to get the old out of the way so that we can scramble our genes, okay, increase the variability of our genes so we can adapt to a changing environment. But you know, humans are supposedly smarter than environment now. Of course, we've made some mistakes, but we've developed eyeglasses, which makes it so that people can now survive even when they can't see. There's a lot of things that we are in control of our own destiny now. But if we had figured out a way to control telomere lengths probably 200,000 years ago, we'd probably be extinct as a species right now because we wouldn't have adapted to changes. Yes? Okay. Who else has a question? I'm going to be around. I'm addicted to talking about this subject. There was a question back there. Yes. No data yet. I plan on doing that study as soon as we get the funding for it. But growth hormone, by definition, induces IGF-1, and IGF-1 induces bone cell and muscle cell proliferation. And the cell proliferation all by itself causes telomere shortening. But I still recommend it. Same is true for mycodermabrasion and immune boosters. All of these things actually cause cell division and therefore telomere shortening. But I have this belief that what's the point of living a long time if you're not living? And I believe human growth hormone is a good thing. And I assure you, I will come to the rescue, okay? Or my lab will come to the rescue and find a way of re-lengthening those telomeres after they're shorter. Yes, any other questions? All right, thank you very much. Yes?